Alrighty, y'all. Final story of the night. This is on a genetic discovery that reveals sex determination in killifish. It is the gene is called GDF6Y, and it's a really interesting system because of how the genetics work. It is not as simple as XY in these fish. So normally, right, sex determination is either XX female, XY male, or we've seen in insects, right, XX female, and then a singular X is male. But here, there is a gene that's in the genome, and it's called the Y chromosomal version of the gene. So this gene acts basically as like the Y chromosome. And what they found is if this gene is turned on in specific cells, it triggers male development independent of whatever germ cells there are. So even if there are ovaries in the animal, which we'll talk about, if you turn on this gene, it will otherwise become male. So it's like the master regulator of that switch that exists. But before we go into some of that story, I thought it'd be important to review why exactly these fish are studied. A lot of the big reason why this fish is studied is for aging research. So aging, we know, is a phenomena that we're all having to hit with right now. Not necessarily analogous to human summer. I think it's super difficult to make the analogy here in them because it's one gene that's acting as if it's the Y chromosome. And that's something that they were able to demonstrate by turning it on in certain cell types. There are animals that are hermaphroditic naturally, like nematodes, but there the sex determination acts in a very different way that's been described. And this is just very, very different to what has been seen before. I figured it would be fun to just take a look at what these fish look like, and even like the location where they're studied and why they're generally studied as well. So they have a really relatively short lifespan. And again, this question here was just trying to identify what is the factor that makes them male and female, since they don't just have a male, like a Y chromosome as your classically defined system, what it would be. So sex determination in killifish is what they were trying to identify. There's no Y chromosomes. They're trying to identify if there is a gene of interest that's acting like a Y chromosome, and that seems to be the case. This one gene is turned on in certain cells and it makes the male development. And when the gene is off, it keeps everything female. And the idea of why it works this way is to keep an equal ratio of males to females in nature. And so it was really cool that if they deactivated and mutated the gene, they made all females. And they were fertile, capable of reproduction. Even if they took what otherwise would have been male, if they turned off the gene, it would be fully fertile female. And then if they took an animal and added in the gene, so it was supposed to turn into a female, but if they added in the gene, it would have a male-like individual develop. Those that was different. So the male-like ones looked and behaved like males, but were not fertile. So that was the interesting part, I think. They also are trying to do a total sex reversion to see, like, if midway through development, like, kind of permute, if you can change the signal of this gene during development, can you start to drive them towards male and then drive them back to female and drive them towards male, and just like at what point can you convert back and forth in terms of that sex determination? But let's just see what they're regularly studied for so far, which is their lifespan. Nature Communications shows still a really good paper and a really good journal of where they published it, but they kind of had like their future goals to see if at what point they're able to turn things on and off and have like a more complete sex reversal. So especially when they did a sex change for the females, where they introduced the gene GDF6Y, there they made the male-like individuals developed, and they looked and act like males but weren't fertile. So that's why they were saying it's not completely a reversal, because they weren't able to make them fertile. But they're thinking it might be, the hypothesis was that the gene wasn't activated in every cell. And so that's a little bit something more they need to look at, is when they're introducing this gene, it needs to be much better in terms of like where its active is. Not as fertile as male. So not fertile as male. Yeah, exactly. So Jay, when you took a female developing embryo and you gave it this gene, this GDF6Y gene, if you did that, if you gave in this gene, that they would go from being female to behaviorally and appearance wise male fish that were just not fertile. And so the question is, okay, why is that? Where in the fertility process did we go wrong? Because what was weird was if you remove the gene from the male, like the male embryo, then the male embryo would become female and it would be fine. It would be totally fertile, no problem. I think the argument that they make is that it's easier to go from removing something and breaking a gene to putting in a different copy, or like the copy that you're trying to put in, which is true. Like that is a harder process of breaking versus putting something functional in. So maybe that more than one gene to make it. It could be. It could be. Yeah. They, like I said, Jay, like they were suggesting that the experimental conditions were not ideal because they weren't able to modify every single cell. And so I think the easiest thing to say is we didn't hit all the genes. If we could hit all the genes in every single cell, maybe it'll work. If you do that, if you are to guarantee to me that every single cell has been modified and it still doesn't work, then it's probably more than one gene, like you're saying. But it is still really striking in terms of like, I think this could still be like the upper master regulatory element, meaning this gene that we have activates and modifies a ton of other things. And so as long as you have that element in, it'll totally go down this pathway. If you have something a little bit further down, right, maybe the pathway bifurcates up near the top that our master gene does. And so if you get something down in the bifurcation, if you modify that, that's not sufficient to do this gigantic change, like to the female embryos making them totally male, right? But if it's high enough, maybe it is functional. And maybe the argument is if you don't change it in every cell, these downstream effectors are also not changed in every cell. And maybe it's one of those that's the critical component, but it would have to be in combination with that upper one, would be my thinking on that. So that's the lab setup. I don't know, it's interesting looking, right? Talking about the master gene prominent. For this one, C-Town, this is considered to be a master regulatory element because it's considered to be really, really high up in the pathway. So it is the gene that is responsible for sex determination in these fish, where the idea is if you remove it, the animal becomes female, and if you activate it, the animal becomes male. Not just in terms of physiology, but behavior as well. And so that's why it's called at that level. Does that make sense? For these, so you have a genetic stock, Jay, the same as we have in fruit flies, where it's like everyone in this container is genetically identical. And so it'll be the same thing for these fish. Like this fish bucket, everyone's the same, just like in this fish bucket, everyone's the same, and so on and so forth. And that is important because imagine, Jay, if we were inserting this gene into different genetic backgrounds and comparing like replicates between those backgrounds, if they all lead to the same thing, then it's more strength to your argument. But if there's an outlier, you have no idea if it's because you didn't do the experiment correctly or there's a genetic element in there that is more important that's blocking your hypothesis, or vice versa. So the way they did it in this study is the best of both worlds because they did replicates of one line of fish and then they did it in another line of fish too. So different genetic background, but doing the same genetic experiment and showing the same results. And that is very, very important to do. So if you can show with the same genetic background, the experiment, then it's going to end up working. Does that make sense? Is one incongruent with hypothesis two? Not necessarily, but the reason they're presenting it in this way is because it's more parsimonious or easier to be like it's one of these two versus there having to be multiple takeaways where it's like more things that needed to happen to make a certain event take place. That makes sense. Yeah, and so that's why I like this paper, Jay, where it was pretty clear cut. Like it was a very straightforward paper, very well done, like very solid science. What would be really cool, Jay? Just think of you in these papers, my friend. Because wouldn't it be neat is if you could attach, like if you're already putting in the gene, right? If you're putting in the gene, the sex determining gene into the fish, attaching a photoconvertible component onto the gene, right? So that you can now activate it with light. That might be a cool way to go. Like if you can make sure every cell is hit. Because actually this group, last year, this group published a paper, not the one in this video, but the one here, they published a paper on they made one of these killifish totally see-through. Like the adults. The adult was totally see-through. And that is what you need for the laser to work, right? So it was behaviorally totally normal. It swam around just fine. It reproduces just fine. So they basically built a tool that they can use. And so now in that super see-through adult fish, you can shine that laser in. And in this case, it would be like this GDF6Y gene for sex determination. And being like, all right, I'm going to activate it and then see what happens. And plan out like where in development it's critical to be able to demonstrate like these changes. And yeah, it's as an adult fish, they made it see-through. Yes. Oh yeah, Jay, like light sheet 2. You want to see the transparent fish? Okay, let me find the citation for it. So look at that. Like, look at how see-through this fish is, right? I mean, you can see the cartilage. You can see the organs. Like that is what do you think, Glued? Jay, like that's a pretty see-through. Yeah. So they named Seatown. They named the fish was named Clara. I believe the gene that they were hitting on this one was like pigment genes. I didn't need you to open it. I think it was pigmentation genes in part that they were hitting. Yeah, simultaneous inactivation of genes involved in body pigmentation. So it was, the Clara was the farm breed. I think it was one of the genes that they discovered in this. Yep, it was the stable line. So it was the lineage that they made Seatown. Basically, you just inbreed them after this. And so, like we were saying with Jay, everyone in that container is now going to be genetically identical. And so they named that mutant line the Clara line. So if you wanted to do this with see-through killifish, you just get a Clara is the name of that line. Just like with a line of fruit fly, well, one is called Oregon R, or another is called Canton S, or another is called Yellow White. Everyone in that container is genetically the same, and here is just like the shorthand name of it. Which I wonder if they have any other images. Yeah, here we go. Look at that. Look at the fins, y'all. You can't see the fins in water. And then you have to take them out of the water to see the fins. Like, this is the normal fish here, right? And then here, like, there's no fin pigmentation. You can't even, like, they're having to give us a little box and be like, this is it. And if you look closely, yeah, can serve as a platform for genetic manipulation. So like we said, the argument is that these are going to be very similar to the reason why zebrafish are so helpful, which is because they are see-through. So if you can start replicating what you see in terms of the genetics and the tools in zebrafish, you can show the exact same thing in these animals that you had to genetically perturb, but now they're also see-through. And that is just, I don't know. I think that's really, really cool. They do mention that there's loss of some eye pigmentation. And in this study, it is kind of like, well, that might have an effect on something. We know eye pigmentation is critical for certain visual cues. And so there might be something that happens as a function of losing some pigment in the eye. Anywho, here is the proving that you can see the glow. So this is seeing through the body, it glows. So this isn't this isn't using lasers to activate anything. It's just showing that you can mark certain cell types and then use lasers to see through the animal and see it light up. I don't know if you need to, they might not have these at a stock center. You might have to ask for them from the company, or not the company, but the lab, because it's so niche. I don't think they have a stock center for it. So a lot of genomic comparisons, figure out what's causing the short lifespan. Meaning, because they have such a short lifespan, their genome has been filled up with mutations that are bad, but a lot of the times it won't hit because they just die so early. So only if you try to nurse them to long life will you start to see there's a lot like the survival of the animals. There we go, y'all. It's a really neat study, right? This isn't the paper that we're talking about. This is just what they're commonly used for. So you have these two populations, one that's long-lived, one that's short-lived. And you could just do the comparison between them. Okay, what does one have that the other doesn't, and try to figure out what causes aging, and then see if those are conserved signals and genes that are also in humans as well. Contribute to the buildup. So because of the short lifespan, they live such short lives that you don't really have as good of a selection pressure, meaning evolution isn't selecting for them to not have these mutations, because they just end up dying before those mutations that'll shorten their lifespan actually end up hitting. And that dying is driven by the fact that the water dries up, right? And so without the water, they're dead. That's it. And so the short lifespan is what's funneling the bad mutations out, like you're saying, in the lineage of it. And if if you try to nurse them, like if you put them in a situation where they're not gonna be in a dried out pond, right? So they can try to, you can, in theory, then they should be able to live longer because you're not getting rid of the water supply. Then all those deleterious or those bad mutations starts to surface, and you'll see like further accelerated aging. By now, yeah. Well, but also that has selected for things as well. So in terms of natural selection, there, what natural selection is acting on is that it's a short season for them to survive. So then the only individuals that are going to survive are ones that reproduce quickly. And so there, that natural selection is at, that's where natural selection is focusing on. You're only passing on your genes if you reproduce and develop quickly. And instead, you're not hitting those deleterious mutations because that's not what you just need to like live fast, live hard, and then die in that situation.